Hi. Our focus in this last chapter of Unit 2 is on the different divisions of the autonomic nervous system along the efferent pathway. Our goal is to have an awareness of the dual innervation of nearly all body organs and the neurotransmitters commonly found in the autonomic nervous system. The autonomic nervous system is built to manage our physiology and maintain homeostasis. It does this by regulating organs and organ systems, including management of the associated muscle cells and glands, which are the effectors we will study more in Unit 3. Autonomic activation of various smooth muscles causes a spontaneous activity of rhythmic contractions like we see in the GI tract during digestion and swallowing. Autonomic nervous input simply increases or decreases the intrinsic activity of smooth muscle contractions. Visceral organs that are under autonomic nervous system control include cardiac and smooth muscles, including arterial blood vessels and glands found in the endocrine system. Autonomic efferent motor control involves two neurons working in tandem along the efferent pathway. This is unlike somatic motor control, which is usually under conscious voluntary control. Let's talk more about Figure 1 in our new chapter. The autonomic nervous system has two divisions, the sympathetic fight or flight division and the rest and digest parasympathetic division. Both divisions will innervate the target effector organs. The primary function is to regulate organs that maintain homeostasis. These two divisions will have antagonistic effects, therefore they do not both function at the same time. They can each be triggered by your activities, and we'll describe some of those here. But hopefully when you hear fight or flight and rest and digest, you can relate to those personal activities that have overriding effects of these two divisions that operate in opposition to each other. For example, when you're running away from danger, you're not really thinking about chowing down on a burger and sitting on the couch. Okay. On the left side of this diagram in Figure 11.1, highlighted in green, the sympathetic fight or flight responses are set from the thoracolumbar area along the spinal cord. So although these responses are autonomic, the central nervous system controls the activation of this division based on perceived danger. Nerve impulses are issued along these pathways. They're highlighted in green. Notice the sympathetic chain, these vertical ganglia that run on either side of your spine, and synapse with the postsynaptic side that runs out to the target organ. This is characteristic of the sympathetic branch, to have short presynaptic paths and long postsynaptic tracts out towards the organs. Now let's look at the parasympathetic map over here on the right and highlighted in purple. This branch controls the rest and digest responses. So when you're relaxing after a big meal, this system will dominate. All the organs of the body are targeted by both branches, but they will not both be activated at the same time. So we say these two systems are antagonistic to each other. Notice the parasympathetic division has these long presynaptic tracts, without the ganglionic chains notable in the sympathetic branch. Here's a closer look at the proximity of the spine to the sympathetic chains. We see these various sympathetic ganglia. They're linked together in these vertical structures like sympathetic chains or trunks. What do you notice when you take a look at the right side of this figure from your book? This is Figure 11.3. On the right, you have effector organs, which one has an unusual setup when comparing it to the rest. It's the adrenal glands. They have a direct connection to the central nervous system and bypass the sympathetic chain. Maybe you can think of the last time you nearly wrecked your car. Think about how fast your entire body responded to the adrenaline effects of the perceived danger. This is a protective device and works almost instantaneously when you perceive a dangerous or scary situation. We'll talk more about this and the adrenals in Unit 3. Here in Figure 11.4, let's compare the pathway for the somatic reflex response. We tried this in lab with the knee jerk reaction. This pathway uses the dorsal root or afferent neural tract to send the stimulus information to the central nervous system, but it only reaches cord level, not the cerebral level, like we studied in Chapter 10 for perception and awareness. Once the signal reaches the spinal cord, an immediate motor response is issued through an association neuron that fires and activates the motor response directly along the ventral root. Usually you don't have conscious control over reflexes. These are also by design protective. Now the figure below, in panel B, we see the autonomic pathway toward a visceral smooth muscle effector. The two neurons in this efferent pathway are the preganglionic fibers, and notice they have the cell body or soma located within the central nervous system. These neurons synapse with the second set of neurons called postganglionic, which are characteristic of the autonomic system. The autonomic system effects on target organs is absolutely dependent upon the type of neurotransmitter released at the target tissue, and of course the type of receptor presented at the different effector organs. It's always nice when we can say always and never, and in this case, all somatic motor neurons release acetylcholine as their operational neurotransmitter. This always produces an excitatory effect, leading to an EPSP. Remember, that's a graded depolarization in the postsynaptic neuron. In the autonomic nervous system, here are some things to remember. Preganglionic fibers release acetylcholine. Those are cholinergic fibers. Acetylcholine is in both sympathetic and parasympathetic branches of the nervous system. Acetylcholine is the only neurotransmitter used in the somatic branch of the efferent nervous system. Postganglionic fibers release norepinephrine or acetylcholine, and the effect is either stimulatory or inhibitory. Neurons that release norepinephrine are called adrenergic. We'll look at some pictures on this as we dive into more details of the autonomic nervous system. Here's a nice simplified look at the parasympathetic branch that has characteristically long preganglionic paths that originate in the midbrain, medulla, pons, and sacral region. That's the alternate name craniosacral division. Out to the right side on this figure, we can appreciate the postganglionic neurons and that they have short axons that innervate or synapse with their target. Remember, the parasympathetic division performs maintenance activities and conserves body energy. It's involved in the D activities, digestion, defecation, and diuresis. The patient's blood pressure, heart rate, and respiratory rates are low, and gastrointestinal tract activity is high. The skin is warm, and the pupils are constricted. The wandering vagal nerve, cranial nerve 10, carries mostly parasympathetic fibers. This is the only cranial nerve that leaves the head and neck area to go on and innervate a variety of organs, including the heart, lungs, esophagus, stomach, pancreas, liver, small intestine, the upper half of the large intestine. We tested this with the Brady test in lab number 5. By contrast, the sympathetic division, also known as thoracolumbar, acts in the fight or flight response. This involves the E activities like exercise, excitement, emergency, or embarrassment. The division automatically promotes adjustments to our physiology during exercise by increased blood flow to skeletal muscles, while blood flow to visceral organs is reduced. The heart rate increases, and breathing will become rapid and deep. The skin may become cold and clammy, and the pupils will dilate to allow as much light into the eyes in order to send maximal amounts of environmental information to the brain. This system uses what's called mass activation. The body systems, due to divergence, will produce a global or total body response, which is often long-lasting. The term divergence refers to the fact that preganglionic neurons branch in order to synapse with a number of postganglionic targets. The global response with mass activation by the adrenal glands, which are found sitting atop both kidneys, is only possible due to the fact that upon direct central nervous system stimulation and the subsequent release of epinephrine, also known as adrenaline, directly into the bloodstream, causes this mass activation of all target organs that have a receptor for this chemical ligand. The ratio of epi and norepinephrine is an 80-20 ratio. But for our learning purposes, we will consider these chemicals as identical twins with the same effect. More examples of using the bloodstream for mass activation and delivery to all tissues at the same time will be explored in Chapter 6. We go back to that chapter at the start of Unit 3 next week. Please study this table from Figure 11.7. It directly compares somatic or voluntary activation to the autonomic system and its divisions. These pathways emphasize the long and short neural pathways between pre- and post-ganglionic fibers. It shows examples here of the target tissues, also known as effectors, and most importantly, at this point, the two neurotransmitters along the way. We can use the key at the bottom of this diagram to remind us of the structural differences between pre- and post-ganglionic fibers. They are drawn with a solid or a dotted line. Most of these fibers are myelinated, which means they will all utilize saltatory conduction, which is faster than a bare wire type of axon. And most importantly, you can see that acetylcholine is used at all presynaptic junctions and is always excitatory. You only see epinephrine in play at the effectors in the sympathetic pathway. Definitely review this diagram before the exam on Monday. Let's look into some details about the neurotransmitters and their tissue receptors. So we'll focus again on acetylcholine and epinephrine. Here in Figure 11.7, we have a similar graphic to the previous one we just looked at, but there is more emphasis here to the right. Here we can see what's happening at the target effectors, and we can see the receptors facing out into the ECF compartment, ready and waiting to catch the signals being released by their postganglionic neurons that are controlling their responses. Remember, most organs have dual innervation. There are a few exceptions, and I'll let you guys read about that from your long version of my slides. We already mentioned the fact that preganglionic neurons always release acetylcholine. The nicotinic cholinergic receptors on the targets are therefore associated with depolarizations, or excitation. You may be noticing the terms adrenergic versus cholinergic here. Let's dive into that next. One way to remember the term adrenergic is, remember the ligand here is adrenaline, epinephrine or norepinephrine, and cholinergic receptors are binding to acetylcholine. We already mentioned that acetylcholine is used at all somatic motor neuron synapses on skeletal muscles and are 100% of the time excitatory. Acetylcholine is also used at all preganglionic neurons, as well as at parasympathetic postganglionic neurons. And you can refer back to Figure 11.7 if you need to. The cholinergic receptors come in two flavors, or subtypes. There are nicotinic and muscarinic receptors available to bind the same acetylcholine ligand, and this depends on the tissue type. With the nicotinic receptor, all subclasses are directly associated with ion channels that allow sodium and potassium to diffuse through the cell membrane whenever they're in the open position. Ions will flow down their established concentration gradients. Remember, the gradients are established in order to maintain resting membrane potential in neurons, but all cells will use the sodium-potassium ATPase pumps to rid themselves of sodium. Nicotinic receptors are sensitive to a naturally occurring compound called curare, and they can become blocked or inhibited when this chemical compound is around. When this happens at a skeletal muscle, that effector is then paralyzed because the depolarization process is blocked by this large, bulky curare structure. Going back to Figure 9.11, we can see the two receptors side by side, and now we can look at their functional effects on the effector cells. On the left, the nicotinic cholinergic receptors bind two molecules of acetylcholine, which in turn pops the ion channel open and sodium rushes in, causing depolarization, and then there is a subsequent loss of potassium. On the right, we can see the mechanics of operating the muscarinic receptor. It's a little different. It only needs one molecule of acetylcholine ligand to operate a G protein style of receptor. These receptors are found on effector organs in the parasympathetic division and will elicit either a depolarizing or hyperpolarizing effect. Our heart, as many of these muscarinic receptors are located, and acetylcholine, which is normally excitatory on all skeletal muscle types, is actually inhibitory when it's used at the heart. When released near the heart's neural synapses, we can have a lowering of the heart rate. Since this is a G protein operated channel, each acetylcholine binds and we see the loss of potassium. Remember from the lab experiments, the hyperpolarization phase, it was always due to potassium channels remaining open after the repolarization phase and the recorded action potential. Hyperpolarization is associated with inhibition. Alternatively, the G protein operation of this muscarinic channel can cause excitation in smooth muscles of the GI tract. Sometimes these contractions can cause a tummy ache or intestinal distress. Atropine is the blocking agent that can be administered in order to inhibit this type of activity. Let's now shift gears a little bit and take a look at the adrenergic effects. Epinephrine, norepinephrine can bind to two different subtypes of receptor and cause different effects in the target tissues. On the left is the alpha subtype and operates under G protein mechanisms through an integral membrane protein acting as an enzyme. Here it's called adenylate cyclase. It's in the middle. In the activation phase, adenylate cyclase burns an ATP and causes a cascade of events through cyclic AMP, which ultimately activates protein kinase. These enzymatic proteins work to phosphorylate cellular proteins that may be in the inactive phase inside the cells. Once this happens, there's widespread activation within the target tissue at the cellular level. The positive and negative signs here inside the phospholipid bilayer, these symbols in the plasma membrane, they indicate activation or inhibition of the cellular response. So we see that the cell can respond to the same ligand, but in two very different ways. This is common, and that's why it's important to go ahead and familiarize yourselves with these different types of receptors.